So welcome back, everyone. I think we're ready to move on to the next session, which is session 5, non-traditional antimicrobials. And I'm very happy to introduce the moderator for this session, Richard Gordon, who's a Director, International Business Development, the South African Medical Research Council, and member also of the JPIAMR Steering Committee. So Richard, the floor is yours.
Hi everyone, it's my pleasure and honor to moderate this session. In the interest of time, I'm not going to give very long introductions to each of the speakers' presentations. I'm just going to introduce the speaker in an effort to have lots of time open for questions. The speakers will be given 10 minutes, and we have 5 very established speakers to come and talk to you. And I'm going to get kicked off straight away with Dr. Tahira Riaz from the University of Oslo, who's going to give us a talk about the development of novel mycobacterial tolerance inhibitors against MDR and XDR tuberculosis. Over to you, Dr. Riaz.
Thank you, Richard, and thanks to the organizing committee for this opportunity. I will be presenting our JPIAMR funded project, development of novel mycobacterial tolerance inhibitors against MDR, XDR tuberculosis, the MTI for MDR TB project, headed by Professor Fredrik Almqvist from Umeå University in Sweden. So just a short background on who I am. I'm a postdoctoral research fellow working on this JPIAMR funded project, MTI for MDR TB. My main background is within mass spectrometry-based proteomic studies to understand macromolecular dynamics in infection biology. I did my PhD within the subjects immunology and microbiology with emphasis on proteomic studies. I would also take this opportunity to mention our national network on turning the tide of antimicrobial resistance. As a scientific vision, I have from basic science to clinical medicine in a translational manner, revitalize existing antibiotics and understand mechanisms in infection biology to control and prevent AMR, which leads me back to our JPIAMR project, the MTI for MDR TB. So just a short background, Mycobacterium tuberculosis is the bacterial cause of tuberculosis disease. TB is the leading cause of death from a single infectious agent and among the top 10 causes of death worldwide. A quarter of the world's population is estimated to have latent TB infection and are at risk for developing active disease. TB is challenging to treat and with extensive and toxic therapy regime and has increasing drug resistance. Tuberculosis patients are given 4 antibiotics as standard first-line drugs, and the Isoniazid, rifampicin, ethambutol, and pyrazinamide. 8% of new TB cases have Isoniazid resistant without rifampicin resistant, while 3.4% of new TB cases have Isoniazid and rifampicin resistant and are multi-drug resistant tuberculosis. In 2019, 10 million people contracted TB with numerous cases of multi-drug resistant TB and extensively drug resistant TB. And a greater understanding of the mechanisms that drive MTB drug tolerance and resistance are needed to improve treatment and to contribute to the WHO Sustainability Development Goal to end TB epidemic by 2030. So, Isoniazid is one of the most effective and specific anti-tuberculosis drugs. It's a prodrug which is activated by the catalase peroxidase enzyme encoded by the MTB-CAT gene. When it's activated, it inhibits the INHA gene and thereby can disrupt the synthesis of mycolic acid. Mycolic acid are essential components of the microbacterial cell wall, and mutations of the CAT-G gene and the INHA promoter or gene lead to resistance to isoniazid. So coming back to our project, focusing on mycobacterial tolerance inhibitors, in short MTIs. In 2019, our collaborators from the Almqvist lab in Sweden and Stallings lab from USA, they published a paper in PNAS describing the MTI C10. In this study, they showed that C10 prevents pellicle formation, which is agglutination of bacterial cells, and thereby made it more tolerant, which make this agglutination makes TB more tolerant to a diverse set of stressors and antibiotics. Blocking pellicle biofilm formation with C10 made MTB easier to kill with isoniazid and curbed the development of antibiotic resistance. They also showed that when MT was grown with C10 plus isoniazid, the drug-resistant mutant bacteria never arose, which can happen in 1 out of 1 million MTB bacteria when they are grown under typical laboratory conditions. So my part of this project is to study the effect of MTIs in clinical isolates. So here is, I'm showing a selection of MTIs synthesized in the Almqvist lab in Sweden. In the first graph over here, you see that there are 3 MTIs, where 2 of them, the one in red and green, have a more potent activity against MTB, again, in comparison to the MTI in blue. We have also tested these in clinical strains with mutations in the CAT-G gene and having high level of isoniazid resistance, and also in clinical strains with mutations in the INHA gene and having low level of isoniazid resistance. What we observe is that these MTIs are more potent in clinical strains compared to the laboratory strains H37RB. So another major part of my postdoc project is to analyze these MTIs and describe their mechanism of action. So some initial studies done on this is I could identify around 2,500 proteins by mass spectrometry analysis with metabolic and cellular processes being enriched, and also enrichment of membrane and intracellular components. I'm aiming to describe the differences and also the similarities between the control samples, which we only treat with DMSO, and then we have another condition where we only treat MTB clinical strains with MTI, and another condition where we have MTIs plus isoniazid, and then a last condition where we only treat them with isoniazid. So this is work in progress where we are studying this, and we are seeing a trend towards that the clinical strains that are treated with MTIs plus isoniazid are more inhibited than the other conditions. So coming to the summary, the mycobacterial tolerance inhibitors, the MTIs, demonstrate the potential to sensitize MTB to stress that is induced under infection. They show potential to improve the function of isoniazid and resynthesize isoniazid-resistant isolates. And we also see similar results in clinical strains of INHA mutant and CAT-G mutant strains, and that these can also be resynthesized with MTIs in a synergistic manner with isoniazid. Currently, the MTI mechanism of action alone and in synergy with the isoniazid and other drugs is being characterized. And I would like to acknowledge our MTI for MDR-TB collaborators, the partners in this project, Fredrik Almqvist from Sweden, Tone Tenjum from Norway, Jesus Blasquez from Spain, Camille Roth and Priscille Prodin from France, and Christina Stallings from USA. And I would also like to acknowledge the people in Genome Dynamics group where I'm working at the University of Oslo and Oslo University Hospital, headed by the PI Tone Tenjum, especially postdoc Siva and Howard for the work with REMA assays and culturing of MTB clinical strains, and also Mari for help with the proteome, preparing samples for the proteome analysis. So that was all from me.
Thank you very much, Dr. Riaz. That's a very interesting presentation. We're going to save all the questions to the end, if we may, and smack bang on time. So thank you very much for getting us off to a great start. The next speaker up is Dr. Mason, Dr. James Mason from King's College London, who's going to be talking about systemically delivered bactericidal antimicrobial peptides to treat bacterial lung infections. Over to you, Dr. Mason.
Thank you very much indeed for giving me the opportunity to talk about our work developing antimicrobial peptides to treat bacterial lung infections. This work is largely a collaboration between King's and Public Health England, and I have a slide at the end of the talk to advertise their capabilities, which are available to you all. So what I thought I'd try and do, and use most of my time, is to try and give my perspective of the landscape for antimicrobial peptide development, and occasionally illustrate how our research fits into the overall field. So in 2016, the Wellcome Trust Portfolio Review considered the potential of various alternatives to antibiotics, and reflecting general disappointment with the lack of success for AMPs delivered systemically, indicated that there should still be some strong support for funding, but it's quite critical to monitor for success in systemic therapy. So moving on to 2019, there's a further review that finds quite considerable appetite for antimicrobial peptide development. There are 27 clinical and 9 preclinical studies, but relatively few were for intravenous delivery, and maybe none of these have direct antibacterial activity. And this reminds us that antimicrobial peptides can be considered a subset of host defense peptides, which often have a range of functions, including immunomodulation. As of 2019, therefore, there were no bactericidal AMPs that could be delivered IV in clinical or preclinical phases. The third review that I sort of promote to you is one from Bob Hancock's lab, and it's quite gloomy, but gives a number of aspects of antimicrobial peptide research that we might focus on to achieve some success. This includes paying more attention to the role of individual amino acids, and also considering that our in vitro testing platforms are unlikely to accurately reflect how antimicrobial peptides act in the infection setting. The suggestion, I guess, is that there's a risk of false positives and false negatives from inappropriate AMP susceptibility testing, and that might be higher than it is for classical antibiotics. And this perhaps captures the most fundamental issue regarding AMP translation, in our view at least, because on the face of it, antimicrobial peptides are exactly what we're looking for. As in contrast with most classical antibiotics, they are produced by multicellular organisms to fight off an infection. However, we rarely consider the environment, including other aspects of the innate immune system where they are produced. It is perhaps underappreciated that other antimicrobial peptides and chemicals produced alongside the hits that we identify, and indeed cells of the innate immune system, may be critical to the activity of our hits when we use them in vivo. Happily, 2020 was a good year for systemically delivered antimicrobial peptides, and there are at least 2 studies published that demonstrate promise. Both studies are notable in that therapy has been achieved with a range of indications, sometimes against gram-negative bacteria, including Pseudomonas, and they use IV, IP, and pulmonary delivery. We also made a small contribution last year, and I'll describe this briefly. In our group, then, our focus is on pleurocidin, and this is a very potent bactericidal AMP. It owes this potency, we believe, to a dual mode of action whereby it not only disrupts the bacterial plasma membrane, like many AMPs, but is also able to penetrate within bacteria to access a range of intracellular targets. Our approach was to modify between 2 and 4 residues to modify the interaction with the bacterial plasma membrane. And one peptide that we found had much greater membrane disruptive activity, and this peptide had also a broader spectrum of activity. What was quite interesting to us is that we observed that for this peptide, where, for example, Staphylococcus aureus was able to alter its metabolism when grown in the presence of the parent peptide, it shuts down fermentation. It was unable to do that in the presence of the new analog. So our modification, therefore, may have fundamentally altered the mechanism responsible for bacterial killing. This particular analog proved effective in our lung infection model used by colleagues in our institute. You may be slightly unfamiliar with this model, but this model involves bacteria embedded in agar beads and uses immunocompetent mice to produce a quasi-chronic infection. The reductions in lung colony-forming units achieved with classical antibiotics are somewhat lower than in other models, but the selected peptide was able to match vancomycin at relatively low doses. So it does look as though we and others have achieved some efficacy with systemically administered antimicrobial peptides. And what comes next? What are the major hurdles for developing antimicrobial peptides as a class? Well, in developing antimicrobial peptides as a class for clinical use, we might consider why they might be adopted in preference to other classes. Is there a USP? Well, it's long been suggested that an advantage of antimicrobial peptides is that bacteria are less likely to develop resistance to these components of the innate immune system, and they've remained effective throughout evolutionary history. This, however, is an open question. On one side, many people highlight the risk that since AMPs might be quite similar to our own host defense peptides, there is a risk of any cross-resistance neutering our own innate immune system, or indeed other membrane-active antibiotics. This has been shown to be the case for example, daptomycin. On the other hand, evidence suggests that when resistance arises, it is frequently much lower than that obtained with classical antibiotics. The changes required for bacteria to have this resistance may affect fitness. And quite importantly, or what we're quite interested in, is that the highly cooperative action of AMPs, which may be enhanced by using them in combination, affords pharmacodynamic properties that might mitigate these risks. It's also possible that small differences in sequence that affect mechanisms of action will also affect the risk of resistance and cross-resistance. So the outstanding questions are perhaps, can the pharmacodynamic properties of antimicrobial peptides really mitigate risk of resistance? Is this a generic property, or will it vary for each peptide, pathogen, or infection setting? So I'm sharing some recent data that perhaps highlights the challenge ahead. It's on the right-hand side of the slide there. You'll note that we're looking at the cooperativity of the same antimicrobial peptide or antimicrobial peptide combination, and it can potentially be affected by the target species. So we've got evidence of a different behavior for Staph aureus versus Baumannii. The culture media, again, that might be different in, we use RPMI and also Muller-Hinton, and whether we're using L or D amino acid peptides. So remember again that although fighting bacterial infections is what antimicrobial peptides are thought to do in the fish or frogs where they've been discovered, we're not evaluating them in the same conditions. So understanding the relationship between PK/PD, activity, and resistance with the innate immune system in more realistic assays and in vivo may help answer these questions and determine a critical feature of this class. So we have recently got some funding from the MRC and Newton Fund. So we're going to look to find new hits with partners in South Africa. And we're going to look to obtain further funding to evaluate as many different AMPs, including those for other groups where they've proven some in vivo activity, according to these criteria. So just to conclude, I'm going to end by highlighting the contribution of our partners at PHE. They offer an open innovation model for scientists from all backgrounds to go to Porton Down to receive training in microbiology techniques relevant for early stage antimicrobial research. They have an established screening cascade for evaluation of novel compounds, including non-traditional therapeutic approaches, which includes a range of facilities such as large clinical strain libraries, the hollow fiber model for PKPD, and Public Health England also have a fully functioning modular hospital ward to test infection control and prevention measures, study disease transmission and assess infection. So you can see on the slide, if any of you have any questions about the model or interested in discussing their projects, please contact Charlie or Mark. So I'll just conclude by thanking all the people that have been involved in the work that we've done and will be involved in going forward in the research that we've suggested as being important. Thank you.
Thank you very much, Dr. Mason. That's again spot on time. Thank you very much. Again, just to remind everyone, for any questions, please can you add onto the chat, and we will come to those at the end. So we're moving on to the third speaker. The speaker is Matthew Upton from Aprologics, and a novel best-in-class agent for prevention of post-neurological infections, AMPND. Over to you, Matthew.
Yeah, so I'd like to thank the organisers as well for giving me the opportunity to speak to you today and to talk to you about some of the work that we're doing at Aprologics. I'm CSO at Aprologics. It's a pre-clinical stage company we spun out of the University of Plymouth in 2018, and I'm CSO there. I'd like to talk to you today about our lead candidate, Epidermicin NIO1, and hopefully to convince you that this is a best in a new class of agent that we're trying to develop at the company for combating gram-positive infections. Taking quite a different stance to that you've just heard about, we're very much focused on topically delivered antimicrobials, and we're doing this because we feel it reflects the nature of the agents that we're dealing with, and also because it will help preserve the small molecule agents for systemic delivery. I assume that I don't really need to introduce MRSA and MSSA to this audience, but I would just like to remind you really that they are still leading causes of healthcare associated infections and infections related to surgical procedures. Also, driving these infections down is a key element of the UK's 5-year National Action Plan, and this demonstrates the relevance of our research programme. Bloodstream infections with MRSA and MSSA are, particularly MSSAs, are on the rise still, and current therapies are failing. More specifically, we're targeting Epidermicin NIO1 for use in nasal decolonization of MRSA carriage. This is a key clinical intervention for preventing post-surgical infections with MRSA. The only agent licensed for this use in the UK is Mupirocin, and this agent has issues with patient compliance, with resistance, and with poor levels of nasal decolonization. Epidermicin NIO1 is a bacteriocin. You've just heard about antimicrobial peptides. Bacteriocins are antimicrobial peptides produced by bacteria, and I'll just point out a couple of differences to the sorts of agents that James was talking about. These are generally narrow spectrum agents, so they target closely related species or members of the same gram class. They are highly potent, probably 10 times more so than conventional antibiotics, and they attack all of the targets that conventional antibiotics do. They can be highly modified, as you can see on the right-hand side here, but Epidermicin is part of the lactacin Q unmodified linear peptide class. These are the key in vitro data that I'll talk to you about today. Epidermicin NIO1 is a 6 kilodalton 51 amino acid peptide, which was originally recovered from Staph epidermidis. And it acts rapidly in a bactericidal manner on the membrane of things like MRSA and VRE at nanomolar concentrations. It's also interestingly active against non-dividing cells, as you can see here, and this differentiates it from conventional antibiotics, and should have relevance in clinical indications where non-dividing cells are present in things like biofilms. Sorry, I'm just going to go. On interaction with target cell membranes, there results rapid depolarization of the membrane followed by ATP loss and potassium ion loss. I'm not showing the data for that today, but the macromolecular synthesis inhibition work we've done with Alex O'Neill at the University of Leeds shows that epidermicin doesn't specifically target DNA, RNA, or protein pathways. And again, this is indicative of a membrane active agent. We've solved the crystal structure of epidermicin and shown it adopts this 4-helix bundle. We've also demonstrated that the structure is very important in the specific mode of action that it employs against membranes. And that's shown here using atomic force microscopy, and we've measured it with other biophysical methods using with collaborators at the National Physical Laboratory. The other thing to mention here is that in all efforts to generate resistant or tolerant isolates of MRSA, we've failed. And this has been shown for other bacteriocins. And although it's encouraging for future potential clinical application, we're not suggesting that resistance will not arise. So based on the nature of the compound, the target organisms, and these data, we decided to employ epidermicin in topical indications, as I say, and our market scoping exercise suggested that nasal decolonization of MRSA carriage was the most logical first market to go to. So I'm showing here the key in vivo data from an efficacy assessment that was carried out in cotton rats. This is a robust model of nasal colonization with MRSA, and that's because when you finish the model, you measure the total MRSA counts in the whole nose rather than just swabbing the anterior nares. In this model, the mice were colonized with up to 10 to the 4 CFU per nose of MRSA, and then they were either treated with epidermicin or with current standard of care, mupirocin. Key point to note here is that a single dose of 0.8% epidermicin was as effective as 6 doses of 2% mupirocin. And eradication of MRSA carriage was seen in 3 out of 5 animals with the epidermicin treatment. This is an unprecedented level of activity in this model and is a major USP over competitor molecules. We feel as though it should be able to lead to improvements in patient compliance and also possibly changes in patient management. So we feel this justified further progression of epidermicin. I should just say here that even with excellent in vitro and in vivo data, we knew that we were not going to be able to commercialize an IL-1 unless we could make it cheaply. And I can't emphasize enough here how the involvement we have with a company called Ingenza has been very important. Ingenza are a leading industrial biotech company based in Scotland. They've worked with a number of key pharma and industry players over the last few years, and now we've started working with them to develop a GMP compliant recombinant expression system, and that's now yielding over 100 milligrams per litre of purified NIL1 at an extremely competitive cost of goods. This system is plug and play and very flexible, and should be of interest to anyone with large peptides or small proteins that they would like to produce. So I suggest you maybe contact Ingenza. This system was developed with funding from the Department of Health and Social Care, and at this point I'd just like to acknowledge how grateful we are for the support from DHSC and other funders for supporting our work. The funding from DHSC also supported development of a final formulation for delivery to the nasal cavity and a full toxicology package. We've received a draft report from this work, and the findings to date are all positive. Micro and macro pathology observations are ongoing. But in a preliminary study of 9 animals, there was a very low level of systemic exposure seen. It was close to the limit of detection, and there were no adverse effects associated with this systemic exposure. The main study was carried out over 14 days in 30 male and 30 female rats, and these were dosed twice daily. And it also included 3 different dosing levels. There were no deaths reported, and no significant changes in weight of either the whole animals or their organs. There were no adverse effects seen in hematology, coagulation, or the urinary parameters that were measured. And although there were some changes in the plasma potassium levels in the animals that received the highest dose, these levels were all within the historical control ranges. So not a cause for concern. We're very aware that a single agent in a single clinical indication is not an attractive prospect for investors. So we're now developing additional programs. This is the nasal program, but on the back of some very exciting preliminary data in the pneumonia field, we're moving into this for a directly delivered to the lung program, which will be progressing in the near future. We're also looking at topical indications in the skin, things like acne and impetigo. And we have an ongoing program of discovery of novel peptides where we're employing machine learning and deep sequence mining to provide new hits. In a competitive landscape comparison, showing at the top here other peptides, there are only 2 that are being developed for inhaled delivery. One of those is now being stopped and another is an immune modulator, so not a direct competitor. I'll just say that we've got a very strong team with all the skills we feel that we need to bring this product to market, and we're optimistic about the chances of success. I won't cover the summary points here, and I'll leave it there, and thank you for your attention.
Thank you, Matt. Thank you very much for the presentation. Again, questions in the panel, please. We're getting a nice little selection of questions going, and we will come back to those at the end. So I'm very pleased to say that 3 talks me smack bang on time, so thank you very much. We're now heading to India, where we have Dr. Grace Cole, who's going to talk to us. She's from the CIHR there, talking about repurposing an antiparasitic as an anti-staphylococcal agent. So over to you, Dr. Cole.
Hi, everyone. Is it visible? Hi, everyone. I am Grace from CIHR, Central Drug Research Institute, India. It is a premier drug research institute with a vision to advance and strengthen the drug development and research in the country. I'm a 4th-year graduate student and currently working as a senior research fellow with Dr. Siddharth Chopra in the Department of Microbiology. My area of research revolves around the discovery of antimicrobial agents against multi-drug resistant pathogens, especially escape pathogens. Today my presentation involves the repurposing of an antiparasitic, which is rather very popular right now due to its potential role in the cure of COVID-19, which is a nitazoxanide against Staphylococcus aureus, currently listed as a high priority pathogen in the priority pathogen list by WHO. So as we are already familiar with, and it has been discussed in the previous sessions as well, that the typical route to clinic for a de novo conventional drug development takes about 10 to 17 years. It's a tedious, long process from lead optimization to be in the marketplace. Over that, if we see repurposing with all its potential substantial obstacles towards success, it still provides a promise to have a fast track to these molecules into the clinical development. Repurposing, which is a strategy to find a new uses for the existing drugs, can of course reduce cost and expedite approval timelines, especially the tedious phase 1 safety phases. We can forego that and directly go to the phase 2A mostly. And since repurposing is relatively nascent in the antimicrobial discovery arena, there's still few molecules which have reached the clinical stages, including oranophen, which is originally for rheumatoid arthritis and amoebiasis. There's niclosamide and antihelminthic now being used as potentially checked as an antiviral, and moxifloxacin, of course, against TB. So for the same purpose, we screened some non-antibiotics against the escape pathogens, and we stumbled upon the nitazoxanide, which is a salicylamide derivative. It is a broader spectrum oral antiparasitic, originally developed in 1970s, and it got FDA approval for the infections caused by Giardia intestinalis, and Cryptosporidium parvum. And it is a prodrug of tisoxanide, which is which is believed to be a non-competitive inhibitor of P4 oxidative reductases involved in the energy metabolism in anaerobic organisms. Pharmacologically, it has an excellent safety profile and moderate absorption properties, and has been repurposed for almost every indication as an antiviral, as an anti-cancer agent, as anti-asthmatic, antimicrobacterial. It reached even clinical trials for Microbacterial, although could not show bactericidal activity there. Currently against COVID-19, it is being projected. Earlier, its activity had been reported when we went into the literature and saw, but there was only initial MIC concentrations research. So we went into the detailed in vitro characterization. This presentation is basically the in vitro data presentation. So when we tried to see the activity against the escape panel, we could see that it had a decent activity against Staph aureus, but not against gram-negative pathogens, which was 16 microgram per ml. But intriguingly, when we combined it with a permeabilizing agent, which was polymyxin B nonapeptide, we could see 4 to 6-fold decrease, and the MICs were near the Staphylococcus aureus MIC, which showed that it might be having a common target and facing the same problem as most of the drugs of the outer membrane of gram-negative bacteria. Further, we determined the activity of Mtis against different clinical strains of staph with well-defined and characterized resistance patterns with some virulence factors such as PVL and different neck cassettes as well. And it had equipotent activity against both the MSSA and MRSA and MRSA strains. So it gave us encouragement to see the bacterial kill kinetics of Mtis against staph. So next we did it, and we took vancomycin as a control, and at 1x and 10x MICs, we checked its kill kinetics. We could see comparable activity with vancomycin at 1x, where here we can see that it was around 1 log difference from the drug-free control. But at the 10x MIC, we could see that vancomycin, of course, was more potent than nitazo, and there was only around 4 log decrease in the 10x of nitazoxanide. So we further saw that if we could see the combination activity of it with some approved drugs, with cell wall inhibitors or the protein synthesis inhibitors, and via checkerboard assay, we could see that it synergized with ceftazidime, gentamicin, and linezolid, which covered both the cell wall and protein synthesis inhibitors. So further, we validated these results with the time kill kinetics assay, and we saw that in combination with linezolid and ceftazidime both, it showed a very good activity. There was around 10 log decrease and 6 log decrease when compared to untreated assay, and just with the drugs alone, there was around 6 log to 4 log decrease when in combined form. So this gave us encouragement for the in vivo studies in combination, which we are doing currently now, but I've not included it in the presentation. And I think nitazo can be used in combination with these drugs. Next, we determined its post-antibiotic effect, which was comparable to vancomycin at 10x. It was 1 hour. Post 1 hour exposure to the drugs, we determined the post-antibiotic effect. So it was comparable to vancomycin. Next, since biofilms are the most dangerous forms of bacteria which it takes and is leading to the most therapeutic failures, we saw its activity against preformed Staphylococcus aureus biofilms and saw that there was significant decrease more than vancomycin and comparable to levofloxacin, and it was able to decrease the preformed Staphylococcus aureus biofilms. Next, which was the most important for us, was to see the propensity of resistance. So we did a study for 40 days where we could see jumps in the Mtis, indicating that there was no stable resistance staph could generate against MTZ, while there was stable resistance for levofloxacin after 40 days. So this showed that nitazo has a potential to go into the in vivo studies, and we could further explore its activity. In the summary, I would like to conclude all the things that I told. It inhibits potent in vitro activity and synergizes with linezolid and ceftazidime against Staph aureus, and it has a post-antibiotic effect of around 1 hour and significant antibiofilm activity. It additionally does not generate stable resistance, which is an important point, and I think it can be used in combination therapy, and we are currently doing with linezolid. Lastly, I would like to acknowledge my funding partners for graduate studies, which is GST Inspire, my institute, CSI CDRI, my supervisors, Dr. Siddharth Chopra and Dr. Anavadas Gupta, my lab members, divisional support staff, and all of you. Thank you very much for including me and for having me to say something at this platform. I'm immensely grateful for all of you to select me, and this has been an immensely informative and very encouraging workshop for me. Thank you very much, and take care, everyone, amidst the COVID wave. Thank you very much. Thank you for the time.
Thank you so much, Grace. That was a very nice presentation. And we are doing very well for time, everyone. So thank you very much. The final speaker in the session, and I know this is a what's the word that I was looking for? A non-traditional antimicrobials, but due to the time difference, we've gone from Norway to the UK to India. Now we're heading west. We're going to Canada, and I hope I'm pronouncing his name correctly, Joseph Abgai, to give us a presentation on host-directed drug therapy in TB. And it's not 100% fit with the topic, but it's better fit with the time zone. So over to you, Joseph.
So my title, as you said, is I'm talking about TB and TB host-directed therapy. The title of this session is non-traditional or unique or novel therapies, but we are humans have innate capability to eradicate TB. 90% of patients or people that get infected with TB can overcome the disease. So what I will talk about is our essays and our options and strategy to try to find small molecules and drugs that can benefit from these characteristics of TB. What we will see here is on the front slide, the first slide, is a picture of called Ophelia of John Everett Maliaris, and it's showing a TB patient suffocating like in water. And the glory of TB is that it is associated with inability to breathe because of it's a lung disease that caused by Mycobacterium tuberculosis. I want to thank the previous speakers that highlighted the disease, but I will talk about our essay to screen and find molecules that can block the replication of TB inside macrophages. So TB is an obligatory pathogen. It lives inside human beings, mainly in cells, in macrophages, dendritic cells, and in granulomas. It's not environmental bug, and it is it needs our the human cell in order to grow. So we decided, and others, to develop assays that will be robust enough and it will be strong enough to characterize and identify novel drugs within the macrophages. It's called ex vivo assay. It was developed within the Tres Cantos initiative at GlaxoSmithKline, and as you see, it's shortened the regular TB CFU assays, phenotypic assay, from 30 days to about 5 days using high content screening, or 6 days using high throughput screening. And we have, as you can see in highlights in this slide, let me move this one. The majority of the drugs are either not working in the macrophage, but there is a big proportion of drugs that are working the macrophages and are not working in vitro in broth assay. So our assay is a high content screening, which either utilize fluorescence or microscopy to read the number of bacteria within macrophages, and thus has the advantage of detecting activity and toxicity simultaneously. We also have developed a robust assay. For that, we use the human macrophages, THP1, which are carcinogenic, but they are uniform and they are replicative from screen to screen, so enable us to screen a large scale of libraries. So our libraries, the libraries that we are screening are either natural product, drug repurposing, small molecules, or focus library. And our ex vivo macrophage infection assay obviously can identify drugs that working on TB within macrophages, either that it's concentrated within the macrophage, and we can identify mutant, and through whole genome sequencing, identify the target within the bacteria, and we publish about it. But the main target of my talk today is targeting the host, which gives some challenges for identifying the target within the host. For that, we use target ID, either omics or use functional libraries. So if a library is focused on small molecules, but a target has been identified for it, we can validate it. However, we also need to address issues of host cell toxicity and range of host cells, because THP1 cells are carcinogenic. Some of the drugs that will work on it will be anti-cancer drugs. So we need to find it on primary cells. And obviously we need to adapt it to animal models. There are some genetic validations for the screening. We use CRISPR, and siRNA, and I will talk about it. I will give two examples which highlight the studies that you do in our lab. The first, we screen the PKIS library. PKIS library is protein kinase libraries of approved drugs and identify leads and hits provided by GSK and University of Northern California. And we started with an assay with a single dose. And what you see here is the activity, phenotypic activity against the bacteria. It means how many bacteria survived within the macrophages, while we can see also that some of the compounds kill the macrophages. So these are in red. The one that kills the bacteria are in blue. And you can see there are several that kills MTB within macrophages with relatively high efficiency. When you look about these families, there are signaling molecules that are target, P38, EGFR. We were interested in GSK3, and GSK3 is a molecule that we have shown earlier to be a target of a secreted protein phosphatase called PTPA. But what's interesting is that if you look at the whole library, there is a large proportion that has some activity against intracellular TB, suggesting that targeting signaling molecules is a valid approach to eradicate TB. So because GSK3 was identified, we went and tested the GSK focus library provided by the BVGH consortium, which shows no activity against in vitro growing bacteria, but surprisingly, the majority of the compound had some activity against TB intracellularly, and these molecules are relatively non-toxic. We selected few compounds, and these shows high activity in a dose-dependent assay, which can even eradicate MTB within macrophages and at a low micromolar concentration. So we went ahead and validated the genetic target, GSK3. It's a signaling molecule, a central signaling molecule. We used several approaches. One of the approaches that we use is siRNA. And GSK3 has two isoschizomeres, GSK3 alpha and GSK3 beta. And when we inhibit RNA of GSK3 alpha, we get about 50%, 40% inhibition. And when we do for GSK3 beta, we get a little bit more. But when we combine inhibition of or silencing of both RNAs, we get over 60% inhibition of intracellular growth of MTB. We further made a CRISPR knockout and showed similar CRISPR knockout against GSK3 beta, because this is the only one that we can do right now. We are still attempting to do a CRISPR knockout of GSK3 alpha in macrophages and THP1 cells. And we showed over 50% eradication of MTB inside of a CRISPR knockout compared to the wild type macrophages, suggesting that the drug activity, or showing the drug activity, is validated by genomics. We can show here that the knockout is completely missing GSK3 beta, and that the drug is affecting both GSK3 alpha and 3 beta. We also identified the mode of action. I will skip how we did identify it, but we showed that we control GSK3 beta, control apoptosis. And when you add the drug, you increase the number of apoptotic cells, which release H37RV from the macrophages. So these are available to be taken out by other macrophages and thus are killed. This activity of GSK3 beta or inhibitors against GSK3 is not limited to MTB. We can observe a gram-negative Mycobacteria, a gram-negative bacteria, Campylobacter jejuni, which shows activity at low micromolar concentration. And we can also block the replication of SARS-CoV-2, which is an intracellular parasite like any virus, by a micromolar concentration of GSK3 beta inhibitors, not the same as one that target MTB and Campylobacter. So this is the effect on other intracellular infection. We also screened in our assay.
Joseph, you have got 1 minute left, please.
I have 2 slides. Yeah, I have 2 slides. With MMB malaria venture, and identified several lead compounds. This is how it looks under the microscope. We have dose dependency assays. It's active against MTB and against Salmonella. This is a work of Enoch Sehil from my lab, who is attending a English meeting, and it's in progress. So our intracellular assay is valid, and we think it's a very useful approach to find new therapies against TB. What we need next is to fill the gap, and we need to raise funding. We need to go through GLP, GMP production, pharmacokinetics and toxicity studies, and go to clinical trial. We already identified a partner in Canada, the Veterinary Institute and Vaccine Development in Saskatoon. And thank you all for letting me present this finding from my lab, and the companies that contributed: Takeda with a Focus library, GSK with a PKIS library, MMB for the repurposed drugs, and Celine, Sandra for the GSK work, and Adrian, and Enoch for the MMB screening. Thank you very much, and waiting for questions.
Thank you very much, Joseph. Could I please have asked for all the speakers to put their cameras on for the questions? I think we have about just over 10 minutes for some questions. And for anyone who is watching them, to please ask your questions. We have had a few, and which have already been answered as well. I think, Matt, you've answered the question, and I think, Jason, you also answered a question on the chat group. The other ones, okay, the questions of spin-outs, And James, so you've answered the question, but maybe if you answered it specifically for the whole group or the private one, but have you observed a correlation between mutants during polymyxin treatment and resistance to AMP? I think you have answered it, but I'm not sure if it is a private answer, but there might be others who would be interested in the answer.
Yeah, I would just say that we haven't actually looked at that specifically. Others have looked at what you get when you adapt E. coli to pluricidin. It's basically something we want to do. And we want to look quite carefully about how similar the mutations are that you get. We predict you will get mutations. From other work our collaborators have done, we've been working with PHE looking at adaptations to antiseptics, and we've been working with people in Berlin also looked at adaptation to antiseptics and peptides, not the ones that I've mentioned in our talk. There is sometimes overlap. But generally speaking, the mutations that we see seem to be quite specific to the particular molecule to which the bacteria become adapted. So I think it's an open question as to what what degree of overlap there is between any two antimicrobial peptides or any agent that has some overlap in its mechanism of action.
Thank you. So I think this question is directed to our first speaker, Dr. Tahira, only because the timing was at the same time you were talking and because I'm not an expert in the area. So it may be from a previous session, but I don't think so. How is resistance propensity, probesnicity for TPP ribose switches? Now, that is the question. It's not really obvious to me, but this came from your presentation, just for the timing. Is that something you can answer?
It was from the previous talk, I think, actually.
Yeah.
Okay. So it's obviously been delayed because it's as opposed to 20 minutes after the previous talks we're finished. So I apologize if that's my mistake. Other questions that have come in, if you just excuse me while I find my way to them before I get to my questions. So I just seem to have lost my bearings here. In connection to host-directed therapies and systematically delivered AMPs, how do we view the potential of exploring organoid technology? Joseph, I think that's for you. And at the same time, there's a comment from Jean-Claude Sirard about some GSK3 inhibitors have already developed for other diseases and entered clinical development. Can these molecules be repurposed for TB? So I think they're both for you, Joseph, and AMPs.
We indeed, you're right. We are testing compounds that are in clinical development. The ones that we are focusing on are from Takeda Pharmaceuticals, which has been explored for treating Alzheimer's disease. So we have a lot of knowledge gained from them. Although working with a large, as a university, working with a large pharmaceutical company has its own issues, but we think we can overcome them. They were very generous in providing us with data and compounds, but we need to synthesize our own to continue the studies. Regarding other microbial agents, I think somebody else, I don't work on peptides, so I think Matt can address it.
Okay, I think it was, okay, so thank you very much. So there's a question here for Grace. ATCC29213 is MSSA. Could you check the activity into MRSA since PB2A is the major determinant of AMR?
Yes, we did check the activity through, I think, 9 strains of MRSA, and they were active for all kind of MEC assets, like 1 to 5 PVL factors, community acquired MRSA strains. So yes, there was equipotent activity against the MRSA strains as well.
Thank you very much. So keep the questions coming, guys. So there's 2 different panels that keep popping up, which get confusing. That's also an old question, so I'll delete that. So I have a question for Dr. Riaz in terms of your clinical isolates, and I think there was also a question for Grace. Where are your clinical isolates from? Are they in the question of yourself, Tahira? Are they European clinical isolates or are they global? And the corollary for Grace, are they Indian isolates or are they kind of global? So where I'm coming from is we need to develop a drug that solves the world's problems, not just India and Norway's. If the question.
So we acquired them from NARSA. They are well characterized clinical strains of Staph aureus, and there was a table included there, and all of them were acquired from NARSA.
Yeah, so our clinical isolates are global, so we have international collaboration, so we get clinical isolates from all over the world. We have especially collaboration with Ethiopia, where we are getting really exciting clinical strains, lineage 7 of tuberculosis, and we have some publications on that too. And other than that, we also have local clinical isolates from Norway.
Thank you. So then the next question is from Matt. So I think you've answered, I don't know if it is privately, but I suppose that's a good general question about the challenge for university spin-outs. You might want to just touch on that for others who are in the same area. But my question is specifically related to the product. I mean, the topical is a very nice application. Is that largely driven by the PK and how quickly an oral might just be chewed up in your stomach? Is that the main driving force behind this, or is that still to be explored?
Yeah, well, just the university spin-out question first, I guess. I didn't answer this privately, but a good question, and one I do come across every now and again. I mean, I think balancing time is a major one for anybody interested in spinning out their technology. Level of support from the university, is your institution particularly encouraging this sort of activity, and if they are they supporting it potentially financially? I mean, to spin a company out and just throw them out there without putting some financial backing behind them is really quite tough, but it's also tough to expect a university to support you. So institutions vary in their rules for doing this and their appetite for doing it, but anybody with an interest, I suggest they talk to their tech transfer office as a first point of call. And then coming to your question, Richard, about topical delivery, yes, I mean it is maybe it's a kind of an easy target. We do feel as though if we leave systemic delivery for other types of compounds, then that does kind of spare up. If we're not using systemically active agents for topical infections that we can cure with things like epidermicin, then that's got to be positive. Epidermicin in particular, because it's not highly modified, is probably more susceptible to some peptidases and proteases than others. Although clearly it's active in the in vivo environment of a cotton rat nose, so it must hang around long enough to have some good activity. So I think it's a balance of being suited to topical application, but also thinking about the balance of sparing systemically active agents for more serious infections, but potentially. Yeah, although clearly targeting pneumonia is a very serious infection, but we still consider that a topical application.
Okay, thank you. And so, Josef, I have a question about the high content assays. What's the kind of throughput of these? I mean, obviously they're pretty expensive to run. Typically they've been the rate limiting step has been the throughput. So as technology and the costs of screening come down generally, just can you give us a sense of what is the throughput? So if somebody wanted to come and collaborate with you, do like 10 compounds a year or 10 compounds an hour? How does it work?
No. We have an automated facility, and we use the Echo Drop for pipetting, and we have the robots, and we can use 384 well format plates. So in a typical screen, let's say for natural products, we can start with hundreds of extracts and then go through fractionation assays. Or if we do libraries, we typically go with focus libraries that are in the range of 100 to thousands, or we can go, we screened already libraries that are in the 10 of thousands within months or so. So it depends on the purity, the amount, and the throughput, but we usually do the fluorescence assay first, and the high content is only secondary. That saves a lot of time. And it's in a BCL3. We do it on TB. We don't use surrogate organisms, so we work directly on TB. So that is done in a BCL3 facility, and this is a limiting factor. Not the throughput. I think we can overcome the throughput. It's the ability to work within a BCL3.
Okay, so I don't have any more questions for the panel, and I don't see any other. Oh, there's another one which I think is for you, James. Do the AMPs impact on microbiota?
We haven't looked at that. It might be one for Matt, actually. You can have that affects the skin microbiome.
Yeah, thank you, James. No, I mean we've not specifically looked either. One of the features of something like epidermicin that we do think is a benefit is the relatively narrow spectrum, so lack of activity against gram-negatives. So skin flora, I mean, I think most of the organisms that we would be, I mean, it's not going to be as disruptive as a systemically active agent that goes into the gastrointestinal tract, I suppose, and maybe that's a positive thing, that positive aspect of the fact that we're not using them certainly orally. So I think the impact on the skin microflora would be of limited significance compared to systemic application.
Great. So I'm trying to get used to this panel where you get asked questions, and when you think now I've got a nice question to ask, it gets answered before I can even ask the question. So Joseph, you were too quick. James, you were too quick, and Matt, you were too quick beforehand. So just as I think I had these nice questions. So my last question is actually for Grace. Nitroxazinide, I mean, it's a drug that's really seemed to be coming back. Everyone's screening it for COVID. Everyone is, it's becoming quite popular. Particularly interested in the linezolid co-activity. You know, so what would be a clinical development of that? I know you're going in vivo now. Is this, do you have a desire to take this all the way to commercial, or to look for partners?
Well, it would be very nice to have partners, but before I would like to see the in vivo activity. Actually, we are trying to either enhance linezolid activity at lower doses, if we can do with nitazoxanide, rather than only focusing on how we can improve nitazoxanide's activity. It's like using a combination. And of course, I would be more than happy if we get partners for that. And I'm actually looking forward to take it forward to commercial uses if the in vivo studies look good.
If I may say something, Grace, we tested nitazoxanide in our assay, and it is highly active intracellularly. We published it, I think, in 2012, and it's actually autophagy promoting characteristics. So you can use about 10 or 20-fold lower concentration in an intracellular assay.
Okay, thank you so much. Thank you. I will definitely try that. Thank you.
Great. So I just, it just remains for me to say thank you very much to the panel. It's been a really truly global panel, going from Vancouver all the way through to India. And I'm sorry, Grace, I'm not exactly sure exactly where in India it is in terms of on the map, looking at it right now.
Lucknow. It's in Lucknow.
Thank you very much, Lucknow. So I'm going to hand over to Mark to close today's session, but I just want to say thank you very much to all the speakers. It's been a very interesting session, taken me about as far out of my comfort zone as I typically can be. But it's very nice to be talking the science again, and over to you, Mark.
Thank you, Richard. It was a really great panel sessions. Thank you very much for the speakers, also for handling time so greatly. So thank you so much. And thanks to the moderators, the speakers, the attendees. I'm going to summarize a little bit this very interesting day, very intense day. So in this second day was really based on research, and we had the first session was session 3. talking about the Mycobacterium tuberculosis antimicrobial pipeline. I was very happy to see that people consider that these are exciting times for TB drug development. There's novel regimens and there's a vibrant pipeline compared to 10 years ago. So this is really great news. We've seen a lot about partnerships and collaborations, which of course are keys to success, not only for addressing the MTB pipeline, but for all the drug discovery pipeline addressing AMR. This is extremely important. Academic discovery, but SMEs, these are agile animals, and they're very much part of the picture. And of course, the pharma expertise needs to be there if we want to really, do more and reduce duplication of effort. I was very pleased to see, and this is really very important as a developer, as myself, I'm the CEO of a discovery company. I think this is extremely important. So when you have your screening cascade and you want to show preclinically that a compound has the potential to become a drug, at the very early stage of discovery, the more the in vivo relevance and the PK and everything you know that could be clinically relevant, and you have ways to measure it, to put it in this screening cascade as early as possible is tremendously important. It can be a game changer. So early hit triage based on predicted in vivo relevance, it reduces drug discovery risk, clinical failure, and of course, all the costs that you will get downstream. And same reasoning for optimal drug dosing. I love to hear when someone said, well, actually several of the speakers said, focus on the lesion, lesion specific PK rather than plasma. It's all about understanding how that drug is going to behave where it needs to be active, right? This is really very important. We've seen novel strategies and outcomes, phenotypic, high throughput screens. We've seen approaches that block efflux pumps, adjunctive agents that can boost the efficacy of antimicrobial products. So this is all absolutely great. I think there's a lot of understanding now that it's not only about showing an effect, but it's also about having the tools to measure and to become clinical endpoints that actually tell you and tell the regulators this is a drug, right? So this is really important. Repurposing of drugs. We've seen the example of nitazoxanide, and that can reduce time and cost, but of course it comes with a kind of new life to an old drug that needs to come with also a set of studies and endpoints that can tell you this is really what we want as a drug repurposed for another indication. Then session 4, novel antimicrobial targets. We heard about the stimulation of the innate immune system as a non-traditional target to quench infection and reduce treatment failure. This is a very promising area, which again comes with also a set of challenges in terms of how you understand resistant development in the context of addressing the innate immune system. We've seen chemical and antibody screens that target biofilm formation. Biofilm is so important in chronic infection and other indications. There is a challenge, of course, of how does in vitro biofilm translate into in vivo, ex vivo activity. This is sometimes challenging, but we've seen a lot of very good ideas today. And other antivirulence approaches also that can be very interesting to avoid gene transfer and resistance. On the novel, and we all love RNA since we have vaccines, mRNA technologies now available to get us out of COVID. We love RNA. And here we heard also that there is very interesting ideas to explore an unexplored area that is the non-coding RNA space in microbes by targeting TPP ribose switches. So very much looking forward to the development of those approaches. And also there was a JPIAMR funded program also that shows us the progress on those novel antimicrobials that are specific against Helicobacter pylori. And finally, we moved on to session 6 that concluded this day of the conference, and I'm looking forward to the next slide. Yeah, session 5, sorry. And yes, I'm getting a bit mixed up. Non-traditional antimicrobials. We've seen traditional, we've seen non-traditional. So there's approaches also inhibiting tolerance. So tolerance, resistance, persistence, they are slightly different things. They all contribute to the persistence of and clinical failure of therapies. Tolerance inhibitors that sensitize MTB to stress and that are induced under infection and improve the function of isoniazid and resensitize infections to the resistance isolates. Also, of course, extremely important to understand the PK/PD, the in vivo relevance, the innate immunity, and the resistance. All these things need to be understood in order to develop antimicrobial peptides that can be delivered systemically. This is, of course, the major challenge for antimicrobial peptides that have worked in other indications, but to deliver them systemically is a roadblock that we still have, and we've seen very interesting approaches to try to break that roadblock. Antimicrobial peptides and also bacteriocides that are some kind of also antimicrobial peptide, they can be very highly potent agents, but we need still to understand what's their exact potential for reduced antimicrobial resistance. And that's, they provide opportunities for directed delivery of antimicrobials, and that could improve patient management and compliance. So a lot of science, a lot of very good science, very breakthrough ideas. Really look forward to see the developments. So thank you very much.